From Microbe TV, this is TWiP, This Week in Parasitism, episode 181, recorded on March 12th, 2020. I'm Vincent Racaniello, and joining me today remotely, Dickson, Despommier.
Hello, Vincent.
And Daniel Griffin.
Hello, everyone.
We're practicing social distancing here.
That's right. We're only allowed to bump elbows online.
We're far away from each other. I'm alone here in Columbia. Dixon's in New Jersey. Daniel's in New York.
Mm-hmm.
But we are here to satisfy your parasitic cravings.
There you go. There you go.
If you like what we do, consider supporting our work, microbe.tv/contribute. Also, don't forget, Parasites Without Borders has a brand new website. Very cool, Parasites Without Borders.
Very nice. Very nice.
And on Facebook.com/Parasites Without Borders. Instagram.com is PWB_worldwide. Twitter.com, PWB_global. All right, Daniel, you've been busy with SARS-CoV-2 and COVID-19, haven't you?
I think that is an understatement. Very, very, very busy. I think on all levels. So that's why I'm looking forward to a little break, getting back to my comfort zone of parasites.
Can I ask you one COVID-19 question? I asked you a bunch last week. We had a special TWiP with Daniel, which was great. Go check it out. To what extent are we testing? Are we still short on test kits for the virus? And should we have more so we know a better idea of what's around?
Interesting. And I'll say today, a few hours ago, this changed. I've been preparing for this for several weeks. I mean, we saw it in China. It just made sense with all the education. So thank you for everyone, all the education I've gotten over the years. I feel like at times I used to joke that, I'm overeducated, but no, actually I feel like I'm appropriately educated. So so I have many roles, right? I'm, one of the directors of Parasites Without Borders. I'm one of the co-hosts on this great show. I'm on faculty at Columbia, so in both the clinical department and basic virology research. But I'm also the chief of the Division of Infectious Disease at an organization, Pro Healthcare. And we have about 1,000 doctors. We have 250 offices, 3 million patients. And so we've been trying to prepare for a long time. And, we thought about this issue of testing a while ago and said, this is going to be a bottleneck. This is a challenge. The CDC is struggling with this. Yep. And so what we did is we reached out early on to one of the big national labs, LabCorp. And basically, we started talking to them. We started collecting specimens early on, even when the testing hadn't rolled out. And then as soon as they had capacity, we started submitting those samples. And actually, as of today, they basically said, we are fully ramped up. We have this workflow established. You guys can send us as many samples as you want, and do not expect delays, and expect us to get the results to you. So at least in this little part of the world, we're really we're ramped up to start testing all the people we need. And I think I mentioned it's been quite a while that Columbia was getting the tests that we needed on the people that we needed. So, kudos to Columbia. I'm very impressed with the people I work with at Pro Health. And you look at what they're doing in Washington, and they're really doing great out there. We've learned from them. We're actually going to roll out a mobile testing, a drive-through testing, hopefully in the next few days, where people can basically line up. We can limit the amount of potential contact exposures. People from the comfort of their car can give us their cell number. We'll be swabbing and testing them, and then they'll be getting a call back or a text back on their cell phone, letting them know the results.
That's great. That's what they're doing in South Korea, right?
That was actually where I got the idea. I think South Korea, and I've been to Alabama, so maybe it was the drive-through experience that I had down in Alabama.
Yeah, that was for a liquor store. So
yes, it was for a liquor store. So
kind of the wrong message to be sending to a drive-through.
I'm willing to learn from everybody. So yeah, just the you get the test done, so we know who's infected, and we know how to address this.
So where will this drive-through be? Somewhere in the New York area or multiple sites?
So we're actually in the whole tri-state area. So we're going to be setting up multiple locations on Long Island, Westchester, New Jersey, even in Manhattan itself. At Manhattan, it'll be a little bit interesting. So we're going to set them up, because people don't want to drive 2 hours. They want to be able to have access to testing in their local vicinity.
Nice.
So,
that's
Daniel,
awesome.
have you seen any actual cases yourself?
Many, actually. And, I think that now it's public. The original case that was identified without, an obvious travel history, we identified ProHealth. And actually, I got the call from one of our primary care docs who saw an individual and basically felt this is just not right. And, I know that I'm supposed to follow these rules, but I'm a doctor and my judgment says something's wrong. That individual is admitted. That individual got tested really as soon as we had access to testing. That individual ended up at Columbia Presbyterian, where he's been getting fantastic care.
Okay.
So great. I've had, I guess, experience for quite a while, and we were seeing cases that were suspicious right about that time as well, and those tests have now come back positive. So sort of, I guess, a lot of experience building day by day, seeing, another case today that we had to admit to the intensive care unit.
What was the tip-off that told the doctor that it wasn't quite normal?
The individual that I think has now been in the press and fortunately been, sort of too much publicity, perhaps, was a younger gentleman, 52, really healthy otherwise, and just the fever was lasting. The respiratory symptoms didn't make sense. All the other testing was negative. Gotcha. And really not having an answer in this context made us say, you know what, this could be the first, the tip of the iceberg of local community spread.
Mm-hmm. Wow.
And kudos to the actual doctor who face-to-face, the primary care doctor who I later ended up connecting with, to say this is not right and I got to do what I think is best for my patient.
Yeah.
All right. Dickson, you feeling well? Okay, everything good?
Background outside, it's not coming from within my apartment.
They're headed for you, Dickson.
No, they're going down.
Well, I hope everyone wants some parasitic entertainment, so let's switch to that.
There we go.
Daniel, it's been a couple of weeks. Remind us what we have here.
Oh, certainly. This is the case from TWiP 180. And this was, I see here, Uganda with a twist. So this was, this is a couple months back now. It seems like just yesterday, but I had returned to Uganda. And as some of our listeners are familiar with, there's a clinic in eastern Uganda by the Kenya border. It's actually up, sort of rising up onto the early part of Mount Elgon, which is where our Marburg outbreaks come from. Speak of another virus. Mm-hmm. And I'm headed to the clinic in the Bududa district of Uganda. But the way I'm going to get there is I fly into Entebbe, which is in western Uganda, and then I'm going to travel by car across the country, and then we're going to head up into the mountains. But I'm actually met in Kampala, actually the capital, because initially I spent a day at one of the local hospitals there, by two young volunteers and Sula, the driver. Actually, he's my buddy who likes to stop for his daily prayers, even though I try to have him bunch them. But Sula is a observant Muslim and actually really, really great guy. But he's fine. But the two women volunteers who meet me, they're not doing so well. And after a little bit of time, they basically say, you know what, can we ask you? And I say, sure, let's. And so it turns out they've been having watery diarrhea, and I'll use the word profuse watery diarrhea, 12 episodes per day, loss of appetite. They don't have fever. There's no blood in the stool. And then we get a little background. They've been living for months at the staff guest house, and they've been doing fine and no problems. But 1 week prior to the onset of symptoms, they went on a weekend trip to an area with waterfalls. And there was this really nice event where they were served an outdoor meal on this just large table with a view of the falls. There was a lot of different food that was served, meats. It was a salad. There was fruit. And then they noticed, as we point out, that they became sick after this. I think I made a comment to help people. I said neither of them recalled that they were served raspberries, basil, snow peas, or mesclun lettuce. So here I am on the spot. I've just met them, and I'm trying to figure out. We're in an area where there's really no diagnostic testing. So I'm going to have to figure, can I recognize these symptoms? Can I offer them a treatment so that we can do an empiric trial of treatment? And I go ahead and I hear this. I say, you know what? I think I know what this is. And I recommend that these two individuals take trimethoprim sulfamethoxazole, or people might know it as Septra or Bactrim. It's a sulfur-based antibiotic. And I say, I want you to take this for 7 days. And within a day or two, there is prompt resolution of the diarrhea. They feel better. The next weekend, we actually go into one of the nearby towns, Mbale. It's about an hour and a half. Well, it took an hour and a half to get there and 4 to get back. But and we go to a place where you can actually get coffee. And one of the individuals drinks basically about half coffee, half milk, and then proceeds to get severe abdominal cramps. So that was sort of our story. I wanted people to see what did they think it was, with this response to my empiric antibiotics, and why did this young lady get those horrible cramps, and why was it my fault?
Right.
Hey, Dickson, take that first one.
Okie doke. Carol writes, "Greetings, Team TWiP. While I listen to every episode, it's usually when I'm in my car, and by the time I would be able to email a guest for the case study, I've moved on to other items on my do list and the email never gets written. Today, I happen to listen to the end of the episode at home, and I'm emailing right away. My guess for the 2 volunteers with frequent diarrhea is infection with Cryptosporidium. My guess is based mainly on the list of food they didn't eat, all infamous for being true sources of crypto outbreaks. My other possible choice would be giardiasis, because I know that there can be intolerance to dairy post-infection, but I'm ruling it out because Dickson had drilled Giardia causes steatorrhea into my head. A quick Google search says that secondary lactose intolerance can develop with crypto, so I'm sticking to my initial guess. Hope I'm right, but even if I'm not, at least I have a chance to win a book. From a sunny day in Victoria, British Columbia, the first one after 3 straight weeks of rain, Carol." Yeah.
Daniel, can you take the next one?
Courtney writes, "Dear doctors, hello from Omaha, Nebraska, where we are currently experiencing a period of warm weather. Hopefully we do not see a repeat of last year's severe flooding. First and foremost, my guess for episode 180 is Cyclospora chiatenensis, based on the 2 symptoms of loss of appetite and diarrhea. This organism is located primarily to the tropics, subtropics, and is linked to unwashed plants such as raspberries, basil, salad mixes, etc. The timeline fits as well, 1 to 2 weeks for onset of symptoms. Another possibility is Blastocystis homini, but it seems this organism usually infects patients that are immunocompromised. Secondly, in episode 178, Dr. Despommier mentions there are no lions in Asia. There is a small population of lions that live in India. This is great. Usually referred to as Asiatic lions.
Dickson.
And we have a Wikipedia link.
No idea. I had no idea. No idea. I take it all back.
There are things Dickson doesn't know. He does have a voluminous amount of knowledge, but there are a few.
It's wrong.
All right. Andrew writes, Kia ora from Pongaroa. Book not one, don't care. The fun of working on a solution is my primary motivation. I keep telling myself that. Weather here is hot, 28°C parched. The whole of the North Island and the top half of the South Island are basically in drought. Bottom half of South Island is being flooded. Climate change is here, and it is nasty. I was one of the many who got caught out last week by the clear malaria test. Lesson learned. False negatives as well as positive exist in the real world and need to be taken into account. Case of the 2 young women with tummy trouble that was cleared up after treatment with trimethoprim sulfamethoxazole. This is what we programmers call reverse engineering. That treatment works for a wide range of conditions, but only a few are parasitic. So I'm going to plump for Cyclospora cayetanensis. Parasitic diseases, lectures number 18, protozoans of minor medical importance. As Dickson notes in the opening comments, minor unless you have it. The symptoms fit as does the treatment. Cramps after milky coffee is puzzling. I assume there might be some lactose intolerance in the wake of the treatment, as the balance of the intestinal flora and fauna could have been disturbed. But I cannot find anything online that would confirm this. So I will put it down to vengeance being taken by the great goddess Caphenia, who is upset at mortals desecrating her great gift to humanity with milk. Namihi, Andrew. Beautiful. Thank you so much. Dickson.
Amanda writes, hello, I am so excited to get my copy of PD7 and it will sit proudly on my shelf when not being read. I do hope it will be signed by the whole trio of parasite professors. On to the case of watery, folly diarrhea. There is no shortage of parasites that wiggle to mind when presented with profuse watery diarrhea. However, I believe these two travelers encountered Cyclospora cayetanensis. The frequency and consistency of diarrhea fit the symptoms of Cyclospora. When the picnic by the waterfall was mentioned, I originally thought of Giardia, but then the treatment would have been some form of flagell. And since they were given trimethoprim sulfamethoxazole, I kept searching the list for likely suspects. Since their gastrointestinal tract has gone through a thorough evacuation, it has not had to replenish the good Samaritans that allow us to enjoy foods containing dairy products, and that is why one seemed to relapse upon ingesting some dairy shortly after her illness. The bland diet and slow easing back into regular fare is the smart way to go. I must cut my email refreshingly short this week, but I greatly appreciate all that you do, and I eagerly await the arrival of my prize. Thank you again, Mandy from Tennessee.
P.S.
Oh, P.S. I'm sorry, P.S. Yes, I had an idea for a new merchandise item, a T-shirt with assorted microscopy photos of parasites on the front and on the back, and with the quote, "You know, it's interesting, Daniel Griffin." A quote from Dr. Daniel Griffin. I have recently noticed that whenever Dr. Griffin says this phrase, which is quite often, some fascinating information is soon to follow. Thanks again.
That's very good.
Nice observation.
Good.
It is, when you listen, you realize all these idiosyncrasies that you don't really notice. And then you realize,
right.
That is my,
But speaking of PD7, one of these days, the two of you need to come here and sign some copies. I have a box of them in my office.
That's true. That is true.
Once COV2 is over, right?
Yeah.
Maybe sooner.
It'll be over.
Sooner.
Daniel is next.
Victoria writes, this sounds like Cyclospora cayetanensis, complicated post-treatment by lactose intolerance as a result of the flattening of the villi of the upper intestine. Her symptoms of anorexia and lack of fever, present in only about a third of cases, as well as the response to trimethoprim, further support this. Most interesting, C. cayetanensis is named for the Cayetania Heredia University in Peru, where much of the initial description of the epidemiology and symptoms was done. Also home of the famed Gorgas course. This is by Victoria Weaver, Adult Infectious Diseases, PGY5, University of British Columbia.
Well, I didn't know that.
So the reason I know this is I actually spent some time at Cayetania Heredia in Lima. And and actually, so I, some of the initial people involved were there. I got a chance to meet them. It's helps to remember things if you go hang out in the areas where they were described.
Dickson, did you ever take the Gorgas course?
I did not. No, I have not. I would have loved to have helped give it, actually, because most of the stuff which they're doing is basic to the science that we're professing on this podcast. So, but I would love to meet those people. I used to know them when they were in a different location in Central America.
Yeah, it was, so it was run for a long time by a guy, David Friedman. Actually, David Friedman set up my Facebook account. So, under the one deal that, okay, David, I'll let you set it up, but you have to friend me. That's what I think people do, or something along those lines.
Right.
But yeah, I actually met him down there. And the funny thing, so many years ago, I had applied to the Gorgas, and I didn't get in, and I was really kind of surprised. So I went to David. I was in somewhere where it was Vienna, Virginia, or somewhere in Virginia. And I went out and I was like, David, what's the story? He's like, you know too much. I was like, that's nice.
Exactly.
But I actually I did do a 2-week expert course there at one point, but it is fantastic. And it's been taken over by an individual who actually, when you go there to do the training, there are medical students who are helping orchestrate, translating, all that. One of those medical students went all the way through and is now running the program.
Oh my goodness!
So it's really nice to see that it's now being actually taught by a local Peruvian who came through the program.
Yeah, it almost went out of business too, and they're lucky they got their funding back.
Yeah, it would be a shame to lose that because it's really a tremendous institution.
I quite agree.
Adam writes, my guess for case 180 is Cyclospora cayetanensis or Cystoisospora belli, based on that they fit the clinical picture and are the only known disease-causing intestinal protozoans I could find that could be treated with trimethoprim sulfamethoxazole. Blastocystis hominis would probably not cause this kind of acute disease. Also, I must tell you that I recently got accepted for a residency in infectious diseases and will move to Skövde, I don't know how to say that, in southern Sweden. A long-time dream is finally coming true. Best regards, Adam. I'm currently in Halmstad, Sweden. Congratulations.
Yes, indeed. Sophia writes, hello, TWiP team. I remember suffering from similar symptoms of diarrhea after a trip to Albania many years ago. I had a similar type of meal, a little bit of everything, including salad, and decided to go see a doctor after 2 weeks of unabated diarrhea. It had never happened to me. I thought that diarrhea always stops after a while. Anyhow, I did get my antibiotics for 7 days and prompt resolution of symptoms. No lactose intolerance afterwards, though. I still wonder what I had, only because I didn't want to get it again. So in this case, I think that the diarrhea is caused by a bacterial agent. The lactose intolerance could be due to Giardia that is also treated with antibiotics. I don't know what Dr. Griffin was thinking. Is Giardia cleared a long time while after the antibiotic treatment? Do the patients need to be reevaluated for a second course of antibiotics is given? Now, it could be that the antibiotics cause the cramps, but this is TWiP, so I had to think of a parasite. After all the diarrhea and the medication, it takes a while for your gut to get back to the normal way, not to mention to get all your energy back. In short, I don't think Giardia caused the diarrhea, but it's the only parasite I can think of that will cause the lactose intolerance. Or maybe I've missed something. Greetings and happy spring, Sophia.
Spring? Where? Is it spring?
It's almost spring.
When does it start? The 21st of March?
21st, yeah, 21st.
Wow.
So we're almost there.
Yep.
Daniel.
Yep.
Renee writes, hello, I'm a longtime listener and a research assistant with Seattle Children's Research Institute. I assist with screening hundreds of compounds and their ability to kill M. tuberculosis. Your podcast played a large part in my decision to pursue microbial research, and TWiP did something even I couldn't do, get my fiance, a mechanical engineer, mildly interested in biology. Now, for the case study, I immediately thought of Giardia because of the post-infection lactose intolerance. However, the patients lacked the foul-smelling diarrhea, and I'm not used to seeing trimethoprim sulfamethoxazole prescribed for Giardia. A quick Control-F through PD7 for everything with watery diarrhea, and trimethoprim sulfamethoxazole yielded Cyclospora cayetanensis. I think this parasite can also induce lactose intolerance in recovered patients. I couldn't find a super clear answer on that. I would order a stool PCR to confirm. Thank you, Renee.
James writes, young woman actually ate food in Uganda and got watery diarrhea. Who would have guessed?
I love that. That's so good.
That's good.
Of Of course, my first guess, totally avoiding the name of the podcast, would be E. coli traveler's diarrhea. Since we use trimethoprim sulfa for E. coli urinary tract infections, perhaps it might work for GI bugs, although, as the Borg might say, resistance makes it futile. Cholera also comes to mind as a possibility here, but this is TWiP, right? The only parasite that immediately jumped to mind when you said trimethoprim sulfa is pneumocystis. But that is now classified as a fairly unique fungus. Shows how old Professor Small is. I thought about Giardia, which is basically everywhere, but I've never heard of sulfa for that. It's an anaerobe, and so metronidazole works. So out came Googleina. I put in parasite trimethoprim sulfa, and up came my guess, Cyclospora. I've never seen a case, recognized a case more precisely. I knew it was a round organism that often stains acid fast. If you think to do an acid fast stain, I don't think it's routine. So if you want the AFB stain, you need to order it. Cryptosporidium can look very similar, by the way, and is also bright red on AFB. Cyclospora can be found luminally in intestinal epithelial cells. Seems it was first described in chimp from Uganda.
There you go.
The milk sensitivity, which sounded a lot like my post-Giardia lactose intolerance, took me once again to the mighty Google. She tells me that several GI infections can make one lactose intolerant for varying periods of time, including a rotavirus, TWiP, Giardia, which caused my kids to call me Mr. Stinky, Cryptosporidium, and Cyclospora. Alexa was of no help. She just told me there's no cure for lactose intolerance and forgot that I mentioned Cyclospora. That's great. James is a professor of pathology at Rocky Vista University, which is in Colorado.
Yep. Cecilia writes, Dear doctors, I think the people living in the staffed guest house are infected with Cyclospora cayetanensis. They are experiencing frequent bouts of diarrhea after their return from a trip where they consumed fresh fruit and vegetables. The above are known to be sources of infection. They were also exposed to fresh water since the area they were visiting has waterfalls. According to PD7, trimethoprim sulfamethoxazole is one of the suggested treatments for Cyclospora infection, and the affected individuals improved after beginning a week-long course of treatment. Cyclospora, like some other parasitic infections, can cause malabsorption. That can explain the severe cramping experienced by one of the individuals after consuming coffee with milk. Thank you again for a great podcast and a learning experience for me and your other listeners. Sincerely, Cecilia. From St. Petersburg.
Peter writes, Dear super spreaders of American trypanosomiasis, in the case study from TWiP 180, 2 travelers who visited Uganda and consumed local food presented with watery diarrhea that resolved after treatment with trimethoprim sulfamethoxazole. This combination, sold under the trade name Bactrim, is recommended for treatment of Cyclospora infection. This infection is caused by the coccidian parasite Cyclospora cayetanensis. This parasite has been described in chimpanzees from Uganda, and according to Parasitic Diseases 7th edition, causes watery diarrhea with 5 to 15 bowel movements per day. Infection is via fecal-oral route and can occur via contaminated food, perhaps in the outdoor meal consumed 1 week prior to onset of symptoms. The sporozoites attach to the epithelial cells of the small intestine, causing disruption that can lead to the malabsorption of liquids. In the case of milk, malabsorption can lead to lactose traveling to the large intestine, where it is fermented, causing abdominal bloating and cramps. This abdominal lactose intolerance should resolve itself as the intestinal epithelium heals over the course of a few weeks. Other parasites considered: Cryptosporidium parvum, Malentidium coli, Strongyloides stercoralis, and Diphyllobothrium latum. All these parasites have watery diarrhea as a symptom, but none of them is treated using trimethoprim sulfamethoxazole. The cure gave the diagnosis in this case. Peter, writing from Cape Town, South Africa, where the mornings are getting a bit colder, but the days remain sunny and warm.
And where I think there are no cases of SARS-CoV-2.
In Cape Town?
Not yet.
Not yet. You think it will happen, Dickson?
Absolutely. It's a big hub for tourists, so I think it might get there like that.
Melissa writes, Dear Vincent, Dickson, and Daniel, giardiasis. If it looks like a duck, walks like a duck, and quacks like a duck, it's a duck. Giardia is one of the most frequent causes of traveler's diarrhea. Other symptoms include cramps, fatigue, loss of appetite. Symptoms of the volunteers check. Giardiasis develops after an incubation period of 1 to 14, which again fits the description in the case. 2 volunteers likely picked it up at the waterfall. Contaminated water or food are common causes. And lastly, cramps associated with milk or lactose intolerance is commonly observed in individuals with recently recovered giardiasis. This tends to be temporary and will likely go away in a couple weeks. Last symptom, check. Treatment recommendation for giardiasis, metronidazole, tinidazole, or nitazoxanide. I'm not super sure why Daniel recommended trimethoprim sulfamethoxazole. Trimethoprim and sulfameth are both antibiotics. Trimethoprim mostly effective against gram-positives, while sulfameth is effective against both gram-positives and negatives. My guess is the volunteers' initial symptoms were too general for a specific diagnosis, so trimethylsulfameth was recommended as it is commonly used for traveler's diarrhea. Thank you, Daniel, Dickson, and Vincent, for all the work you put into this amazing podcast. Here's an alcoholic beverage that I think Dickson might particularly enjoy. He sends a picture of the tapeworm shot.
I see it.
1 ounce vodka, 1 ounce mayonnaise, dashes of Tabasco, and black pepper. I guess that's the mayonnaise curling up in there, right?
Let's hope so.
Sure looks like a tapeworm.
Unless you get closer, it actually looks rounder than it does flat. So I don't know. I'd be suspicious.
So you'd be okay.
It's an albino asterisk.
Very nice.
Yep. Let's see. Western University Global Health Track students Dana, Stephen, and Chris write, Hello, TWiP professors. We believe the most likely diagnosis to be Giardia given their high number of diarrhea episodes and subsequent lactose intolerance. Persistence of symptoms and lactose intolerance could last for weeks to months following infection, so the patients may need to be retreated with metronidazole. Differential diagnosis would include Cyclospora cayetanensis, Cryptosporidium parvum, and Entamoeba histolytica. Sincerely, the Western University Global Health Track students, Dana, Chris, and Steve.
Daniel writes, how do I pronounce that first word from my Italian co-host? Ciao. Ciao hosts. We actually have a large Italian population in one of the hospitals I work at, so I'll have to greet them this way. Now that I have learned this. Greetings from Italy. I'm a PhD student at the University of Padova, studying circadian rhythms and immune responses in insects.
Interesting.
I think that is very interesting and timely. The weather has been fantastic without a cloud in the sky all year. I'm living in Veneto, a region badly hit by COVID-19. And though the university has made great efforts to stop the spread here by canceling all classes and any form of students or staff grouping, we hit the 1,000 patient mark this weekend in Italy. However, today I didn't see a single person on my cycle to the lab. Why? It started raining last night, so there may be a simple solution to the problem. Anyway, sorry, wrong podcast. For my guess, no, I appreciate the update, actually. For my guess regarding the case study, I slipped between 2 pathogens, Cystoisospora belli and Cyclospora cayetanensis, both of which causes watery diarrhea and where the infection is typically caused by fecally contaminated food. Both are treated with trimethoprim sulfamethoxazole. Daniel mentioned specific foods, for example, raspberries. And looking through the PDF of parasitic diseases, I think Daniel was thinking Cyclospora is the culprit. So I'm guessing the 2 women had a tough bout of cyclosporiasis caused by C. cayetanensis from fecally contaminated food. Thanks again for the amazing work you all do. I love the podcasts, all of them, and you keep me sane during my lonely PhD lab work and also during this crazy virus lockdown. I won't stop guessing until I win a book, and even then I will continue. All the best, Daniel.
What virus you work on, Daniel? Tell us.
Yeah.
You could, or you could write into TWiP, but here, viruses are parasites. It's fine.
Absolutely.
Okay, I have Kevin.
Yes, you do.
Disinfection.
This dissertation.
Disinfection, decontamination, fumigation, incineration. That ought to teach diarrhea a lesson. 2 young female nurses, Uganda, 7 days post-countryside tour, diarrhea, watery, severe, no blood, prompt resolution with TMP SMX, post-diarrhea, lactose intolerance. I'm spending so much time listening to TWiP for coronavirus updates that I've been neglecting answering the TWiP 180 case. I will be uncharacteristically brief. Our afflicted patients have a foodborne illness, specifically protozoa, gastroenteritis with diarrhea complicated by a post-infectious functional gastrointestinal disorder. This is basically a case of traveler's diarrhea, which we discussed on 2/27/2019, TWiP 166, presented on TWiP 167. So since our focus is parasitology, we can dispense with the many bacterial and viral causes of gastroenteritis. Helminths, such as strongyloides, may cause diarrhea, but the clinical scenario here is not consistent with a worm infection. The quick resolution with trimethoprim sulfamethoxazole strongly implicates cyclospora as the culprit. A 2017 prevalence study of protozoal infection in western Uganda showed that 36% of their sample, N equals 200, had protozoal infection and 3.5% had cyclospora. Consideration may also be given to cystisospora infection, which is also treated with TMP-SMX. Emergence of gastrointestinal symptoms with milk consumption after resolution of the acute diarrheal illness is consistent with acute post-infectious functional gastrointestinal disorder, here manifested by lactose intolerance due to hypolactasia, transient deficiency of intestinal lactase. This will likely resolve after several weeks, but chronic GI disturbances can occur and evolve into post-infectious irritable bowel syndrome. All patients with a resolved acute gastroenteritis of whatever etiology should be counseled on dietary discretion, such as avoidance of dairy for 2 to 4 weeks, avoidance of fried food, greasy food, and decreased consumption of concentrated sugars and indigestible carbohydrates such as sorbitol and xylitol. Well, that pretty much rules out all food, doesn't it?
Yep.
In the interest of completeness, it should be mentioned that whenever symptoms change or remain in a treated gastroenteritis patient, persistent infection, co-infection, reinfection must be considered. Relevant supporting references are in the endnotes. Thank you for your ongoing efforts. And I should remind listeners that if you go to microbe.tv/TWiP, you will see the show notes for the episode, and at the bottom it says letters read on TWiP, well, in this case, 181. And you can read all of Kevin's endnotes, which are extensive, references, diagnostic considerations,
amazing
tests, post-infectious gastrointestinal disorders, and finally, a terminal curiosity, which I always like to read. One of the thrills of being an altar boy in the 60s was serving at a funeral mass. There was a great deal of medieval ceremony, the chief one being the burning of incense. The censer was attached to a long chain, and the priest would slowly rock it around the deceased, letting out great clouds of smoke that drifted up to the rafters. The aroma of frankincense was transporting. Recent international television footage from coronavirus-stricken areas showed workers in protective gear wielding fire hose-sized equipment, letting out massive plumes of unknown vapor. I figured that this ritual was meant to assuage the populace. Speaking of medievalism, the whole idea of fumigation recalls the Black Death, beaked masks full of herbs, self-flagellation, and animal sacrifice. My immediate reaction to fumigation in relation to the current coronavirus outbreak was extreme skepticism. Then I doubted my skepticism, prompting the question, what is the role of fumigation in epidemic virus infection control? Fumigation is currently used in grain decontamination, rodent control, and in some animal husbandry applications, foot and mouth disease control. And fumigation was used successfully in the inactivation of anthrax spores in the post-9/11 bioterror incident, also known as Amerithrax. Nevertheless, multiple literature searches in PubMed and general Google searches using a wide variety of search strings did not turn up any useful references that describe the efficacy of fumigation for surface environmental contamination with human disease-causing viruses. My tentative conclusion is that fumigation in coronavirus control is reminiscent of medieval plague practices. I remain open to contradiction. I would agree with you. I think cleaning the floor is not going to help unless you have a baby that's going to crawl over the sidewalk, right? But all this fumigation of sidewalks and signs,
it's
medieval.
Yeah.
Anyway, thank you very much, Kevin. Lovely. That's it. That's it for our guesses.
Yep.
Well, no, we have two more people that haven't guessed.
We do. We do have two people. That is true.
Dickson, after you.
After me? Well, I would agree with the majority opinion on this one. I think unless I could see a microscopic examination for my final diagnosis, I think the fact that it resolved with trimethoprim sulfamethoxazole, and it's in a hotbed area for the transmission of Cyclospora creatinensis, rules that in. And I think by presumptive treatment, you've got your diagnosis in reverse, basically. Although I agree that some other possibilities exist as well, and it could be bacterial as well, but probably not for that long a period of time. And watery, no, that's probably Cyclospora. So I'll throw my hat into the Cyclospora ring.
So I initially thought Giardia, but then there was the absent Diarrhea, right?
Right.
And then the treatment didn't seem to make sense to me. So I looked further and then realized that Cyclospora was a common cause of these symptoms. And then I found that you treat with it. And so I would guess, but I'm not entirely certain. I have a little uncertainty. So I'm awaiting the voice of an expert.
Well, I'm glad you say, you have a little bit of uncertainty, because that's the difference between practicing in a limited resource setting versus practicing in an area where you can actually have confirmatory testing, or you can even have testing that then tells you, even if you're not sure what to do. And in the United States, in a lot of centers, we do these sort of syndrome-specific testing panels. And in Kevin's extensive email, he actually puts in a couple of these gastrointestinal pathogen panels. And so, when I trained, it very much was, what I find is really helpful in a limited resource setting is I would go through like, okay, you have diarrhea. Let's talk about the diarrhea. People look at you like, okay, we're going to spend 10 minutes discussing my diarrhea. How often is it? What does it look like? And you can go down that road, and which is what I did in this situation, and said, okay, let's talk about potential exposures related. Okay, I feel like maybe there was a 7-day gap here, so I'm thinking about incubation of 7 days. Tell me about the diarrhea. It wasn't bloody. It wasn't greasy or steatorrhea. It was watery. Exactly. A little bit of diarrhea, a lot of diarrhea. It was 12 times a day. Okay, a lot of watery diarrhea. So I start going through like what clinical syndrome is most consistent here. Now, in a lot of settings, we say, you got diarrhea? Let's do a stool GI PCR panel. And that will look for, with really high, like 98, 99% sensitivity and specificity for about half a dozen different bacteria that are sort of common bacteria. It'll also look for different subsets of E. coli. It's going to look for parasites. It's going to look for viruses as well. So, there's a little bit of a change about how we practice in the States. So here we're saying, okay, what do I think it is? And here I've got to, impress these volunteers, or they're just not going to listen to anything I have to say from here if I can't fix them. And then the therapeutic trial. If I thought it was a bacterial traveler's type diarrhea, in a lot of parts of the world, we've switched from Cipro over to azithromycin. When we're thinking other things like here, where I was actually thinking Cyclospora, we go with trimethoprim as a therapeutic trial. And I expect within 48 hours to see a really significant impact on the diarrhea. And if I didn't treat them, the diarrhea might actually go on for another couple weeks. So it's really, you're looking at a long duration, watery, sort of debilitating how you function when you're having diarrhea, 12 times during the waking day. So yeah, that was the way I went through it. And I always like to pick cases that I think have, I think cute little interesting twist to them. I thought it's nice to have a cyclospora case in Uganda, right? That's sort of, because it was described
Right.
there in the chimpanzee. So I hope people enjoyed that little thing.
Sure.
And the last part, this is maybe something hopefully clinicians will learn, because I've run into this a few times. And in this case, it sort of caught me. Where I was in Uganda in the Bududa district, there was an outbreak of hoof and mouth disease. So there were no dairy products. You were not allowed to buy and sell cows. Cows couldn't be brought to the market. You couldn't drink any milk, and so there were no cheese. But then when we went down into Mbale and left the zone, the Bududa district, and went where there actually was milk and cheese, it didn't click to say, oh, by the way, now that because this, we were in a dairy-free zone. And of course I sat across the table while this individual had a nice big serving of milk. And this is the post-infectious lactose intolerance that several people described. And when she started having stomach cramps, I was like, oh, I'm so sorry. I forgot to mention that. I didn't realize we'd be heading to a place where there's milk. So.
That's funny.
Right. So there is no microscopic diagnosis. So you could possibly have chosen Cryptosporidium parvum, but that doesn't usually respond to drugs. So, and it's usually not as prolonged in patients that don't have immunosuppression of any sort. So I would totally agree with the consensus diagnosis.
Yeah, let's go through the others. So that would be the distinction there, where Cryptosporidium is not going to. Well, the duration I think is a big difference. It's not going to go on this long. That's the same also with most of our bacterial and our viral diarrhea. They're not going to continue this long at this really high rate into healthy individuals. And I think we can also, the healthy, we'll say with the cytoisospora, is that also is something that we're not usually seeing this degree of diarrhea in healthy individuals. The fact that there wasn't steatorrhea and that bloating, though, there's certainly a high amount of Giardia in this region. It just didn't fit clinically.
Right. And someone guessed Entomia histolytica as a possibility, and of course there was no blood in their stool, so you might rule it out just based on that. But
that's good. That's good that you bring that up, because sometimes the blood can be microscopic. We didn't test that, so that's also another possibility related to the duration, but maybe not so much diarrhea. So it was, so there was a certain point when it was like, okay, this is a therapeutic trial because I don't have, confirmatory. I don't have a lab here that could do this kind of testing, and then seeing the response to therapy. And when I saw the rapid and effective response therapy, that really sort of adds a confidence to, okay, I think we know what we were doing here. But as I always point out, that's great, but in a perfect world, we want those labs. We like that self-correction, so we feel like we keep learning and we feel like we're getting it right.
Oh well.
All right, and everything was well.
Everything went well, and both individuals did really well. And actually, one of the individuals is already back in the United States, and second individual is actually about to depart Uganda, maybe, if they can make it back.
Maybe, exactly. To go where?
Heading back home to the Boston area. So
okay.
Dickson, do you know?
Yes.
Or I should say, do you know a very well-known virus that was originally isolated in a district of Uganda, shall I say?
I do. It was in the West Nile district.
I was not going to say that, so I wouldn't give it away to you.
No, it's the West Nile. It's the West Nile virus, and absolutely right. And that's absolutely true. And you remember what they mistook it for in the beginning, too, right? They thought it was a case of trypanosomiasis, but it turned out not to be that at all.
Interesting.
And so, it's a let's say a crucible of evolution of infectious diseases. How's that?
It's where also the Zika forest is located, right?
Right. Exactly.
And apparently cyclospora.
And they had a big outbreak of Ebola, as I recall, too, no?
Mm-hmm. Oh, yes. They've had multiple Ebola disease outbreaks. Yes. Okay.
And Daniel identified a mountain as the source of Marburg virus. I didn't know that.
Yeah, well, of course, it was first isolated in Germany, which is why it's called Marburg, exactly, Germany.
Exactly.
But originates from primates imported, non-human primates imported from Africa.
Right.
Let's give away a book.
Indeed.
A book which has yet to be signed, but many of you are waiting or backlisted or what is it? Waitlisted. Yes. We will get the books out.
That's right.
We will get the books out. So let's see. We had 12 people. Well, we had more, but as far as I could tell, the others had already won, like Kevin. And the people who sent me their addresses, obviously, they've already won. So let's pick a random number between 1 and 12. Are you ready?
We are.
The number is 10.
All right.
Let's see who's got number 10. Melissa, who said it was Giardia, and she sent us the tapeworm shot.
That's okay. No. Well, she gets 2 extra points for that one.
Winner. So, Melissa, send me your address at TWiP@microbe.tv. If you are in the U.S., that's all you need to do. If you're outside of the borders, you could send your address. Not your address, obviously. I need that. Your phone number, which I need to put on the package. And I want to know, is that mayonnaise or tapeworm?
Or, you know what else it could be mistaken for is a bunch of mealworms, very large mealworms. So I don't know.
To me, it looks like noodles, pasta.
Yeah, well, that could be too. That's absolutely right.
But why would you put that in a drink?
I wonder what it tastes like.
Well, we could make it.
Who would drink it?
Well, without the tapeworm or whatever it is in it. Tell us, Melissa, what it is. Very nice. That was lovely. That could be our episode art. We have a very cool paper suggested by Dickson, published in Science Magazine. It's really cool. Do you like this, Daniel?
I guess I was going to, I want to avoid my mannerisms that have been pointed out. I enjoyed this. I've been, I think, working really long hours, and I got a chance to sit down and read this, and this was fun. This was a fun.
Do you have to go back to work tonight, Daniel?
I pretty much work in 24/7. It's long days. After this, I'm going to be talking to Amy about what we discussed earlier, trying to work through protocols about which medicines we can hopefully start using in controlled trials to find out what really works in COVID-19. So I'm going to be working on that later. And there's a lot to do here.
But you don't need to go back to the hospital, right?
No. I have tonight off, and then tomorrow morning, starting at 8 a.m., I work straight through till Monday evening.
Oh my gosh!
So sounds like you're a resident.
No, they get to go. They get to go home.
Well, all right. Well, we will try and get through this paper and let you go. But it's cool. It's called "Engineered Symbionts Activate Honeybee Immunity and Limit Pathogens." And this comes from University of Texas, Austin, where there is the very famous Nancy Moran, who loves to work on these sorts of things, very well known for endosymbiosis studies. And here, the first author, Sean Leonard, and she is the last author, Nancy Moran. All right, so we all know from TWiP that honeybees are experiencing high colony mortality, which is thought to be in part caused by viruses and mites. The Varroa destructor mite. Love it. And so here they want to solve this problem. And the basis for this is RNA interference. And in insects, the cool thing about insects is that in insects and also in plants, RNA interference is a definite antiviral defense. For sure. There's a whole machinery to generate interfering RNAs from incoming viral genomes and to then use it to target infecting viruses. But what you can do in an insect cell is you can actually add double-stranded RNA to an insect cell. You could inject it into a honeybee, and it will block virus infection. It spreads systemically. It's very, very cool. We have talked with Raul Andino about this on TWiP. His lab does this. But they say here, it's not really practical to inject small RNAs into honeybees in the field. It's not even easy to feed it to them because it's transient.
How about just the queen bee? Why not just her and let her do the rest?
Well, Dickson, that's above my pay grade. I don't know.
Just raising that as a question.
What do they? They feed everyone?
Yeah.
So you could do the queen.
They give rise to the entire hive.
So you could give her something to make continuously double-stranded RNA. Yeah, that's a good idea. Using the technology here, that's a good idea.
Exactly.
Maybe that's what they're thinking.
Have to inject it into one animal.
Yeah. So these honeybees have a bacteria. They have a gut flora, of course, and one of the bacteria is called Snodgrassella alvi. Wonderful name, Snodgrass. And they are, in this paper, manipulating this bacterium to continuously make double-stranded RNA directed against the virus and the varroa mite. And it works. If you want to stop listening now, it works.
No. There's more to this. Come on.
They take a plasmid, which is a circular piece of double-stranded DNA. They put a GFP gene into it, and then they put it into snodgrass, and they have now snodgrass that are green, and then they feed this to bees. Treat bees with antibiotics, right? And then they inoculated them with a plasmid.
Right.
With S-alvi transformed with the plasma. They inoculate them with the bacteria, which then grows in them, and it glows green, so they know that this thing can become established in them. That's the key here. So they treat them with antibiotics first to get rid of their gut flora. They put this in, takes over, and it lives for their lifetime, which it's not very long.
You know, it's interesting, the wording, right? I mean, feeding, right? I mean, so they use the term we inoculate, and then they refer to this Snodgrass alvi as actually being a bee gut symbiont. So by inoculating it, sounds to me like it was feeding. It sounds to me like feeding. I mean, that would have been a little, because in these papers you don't have like a really nice method section, so it's sort of it's written in. But that's what it sounds like. If you're going to get a gut pathogen into a bee, that's you feed them.
All right. So just putting this plasmid in the bacterium, you put it in the bees, the thing persists. It's in the gut wall, and it stays for long. They look at it for 15 days. How long does it says persist stably throughout lifespan of bees reared in the lab? I don't know how long that is, but it must not be a long time, because they have to sacrifice them to look for the production.
Well, yes, but I think bees do. I would bet a month would be a long time for the bee to stay alive.
I mean, so bees live longer than other insects, which is interesting. So they actually live, some bees actually live for months, 5 months, like some of our honeybees here. So I don't know the lifespan of this specific bee. Actually, I should look that up.
Well, it's the queen. I think there's only one species that we use here, and that's Apis mellifera.
Yeah.
Yeah, do you want to, can you Google that? Because I can't even spell.
Sure. I can. No, it's right in the first sentence of the article.
you can spell Apis mellifera. It shows,
by the way, just as an aside, this bee has been depicted in hieroglyphs in Egypt, dating back thousands of years. So we've known about honeybees for a long, long time.
So the lifespan of a bee depends on the type of bee. Drones live 8 weeks. Workers live 6 weeks during the summer and 5 months or more during winter.
Fair enough.
But the queen must live forever. For a long time, the queen lives, right?
The queen lives a long time, but...
that's why we say, long live the queen.
That's
right. Exactly right. Exactly right.
Anyway, so now the next thing they do is they engineer a plasmid to make double-stranded DNA. So they put 2 promoters pointing to each other on the 2 strands of DNA, and they're each going to make a single-stranded RNA, which will hybridize, right? And they put that into the bacteria. They feed it to the bees, and they have a GFP on this plasmid so they could see where it is. These plasmids make double-stranded RNA, and they actually the double-stranded RNA is antisense to GFP. So they can detect that. And they can see this RNA being made in the hemolymph, the heads, where there are no bacteria. So the RNA is being produced and getting out of the cells and going elsewhere. And then they also notice that other genes are upregulated in bees that are related to the RNA interference pathway, like helicases and so forth. So, all right, so they can make double-stranded RNA in bees for a long time, and it seems to circulate in the hemolymph of the bees. So that's good. And so next, can they silence genes? So they pick a gene in the bee called insulin receptor gene, and they make double-stranded RNA that would target that. And they see, after inoculating with the bacteria, reduced expression of that gene. So looks like it works. How about pathogens? So one of the viruses responsible for this colony decline is the deformed wing virus, which is a lovely name. It deforms their wings, right?
And what kind of virus is it?
It is a Iflaviridae. Ifla, that's the family. It's not a picornavirus. It's an Ifla virus. Okay. Anyway, so they make a plasmid that will make double-stranded RNA that targets that virus. They inoculate the bees with the bacterium. They let it establish. Then they inoculate the bees with the virus, and they look at replication 48 hours, and they see that it reduces virus levels and it increases the survival of the bees, which is good.
So, Vincent, is there a toll-like receptor story involved in this protection at all?
Yeah, bees do have toll-like receptors, and it is involved.
Double-stranded RNA, would that also trigger the toll-like receptor immune system?
That's a good question. I don't know the answer.
I was going to say no, right? I feel like this is the whole dicer RNA interference pathway that they're capitalizing on.
They do talk about that. That's true. They do.
Yeah, this is a separate pathway, which is before the innate immune response. But there are toll-like receptors, and they have some role in microbial defense, but I don't know what it is, frankly. Sorry.
Okay.
But no, I like where we've gotten to here. I mean, the last two figures sort of bring it all together. That does this work? Can we get this to work? And then do the honeybees do better? Can we improve honeybee survival? Yes.
Right.
Right. And then and then they actually go after can we kill the mites that are feeding on the honeybees? And they can. They do the same thing. They put the double-stranded RNA of a mite, some genes of mites that are known to be essential for survival, and it works. Yeah, the mite loads are lower and the survival is increased.
Exactly. There's a lot of stress in honeybee management because they're shipped all over the country at various seasons, right? But they're stored in places like Texas and Arizona and New Mexico, where the temperatures favor their survival. They sort of go into a dormancy period, and I guess that's why they can live for 5 months, or rather than the short length of time that they live when they're active. But it's a very, very lucrative business for the people that are managing these honeybees.
That's right.
Absolutely, and to see the loss of these would be tragedy for a lot of industries that revolve around agriculture.
So the two questions I have, the one you raised, Dickson, can you feed the queen and will she pass it on? That would be interesting.
Yeah, right.
Will she pass the symbiont on, the bacteria?
Exactly.
And the other is, if not, do you have to feed it to all the other bees? And if so, how do you do that? How do you feed bees? You put it in their honey?
Yeah, you do, actually. You do.
Yeah.
The honey is consumed by the growing larva.
Mm-hmm.
And they could acquire it that way. You've got the same problem, I guess, with Wolbachia, getting that into various insect vectors and trying to see what effects they have on disease transmission.
Yeah, this almost seems easier because it looks like you probably just spray it on the food, and then it ends up. I don't know if people ever brought up this book before. This is what, as I was reading this, I was like, this reminds me of a book I once read. And the reason I read it really was initially because of the title, but then I was like, I actually really enjoyed this. A book called Parasite.
Yes.
So it's written by Mira Grant, actually Seenan McGuire, who she actually has a copy of PD6. I need to send her a PD7 because we've emailed, and I like her book, and I don't know if she likes ours. But it's what they do. What happens in this story is they're using they take tapeworms, and they actually genetically modify the tapeworm, so the tapeworm is producing on a constant basis whatever is needed for like particular medicinal issues. And so it's kind of the same thing. Like instead of trying to put a gene into a person, put a gene into a honeybee, you put it in something that's going to exist in the gut for a period of time, and then it's going to be secreting it. And then basically, you get your hypertension medicine secreted to you by your tapeworm, or here the bees are getting their special antiviral therapy from their gut microbes. And so,
right. It's not a bad idea.
They call this symbiont-mediated RNAi.
Ha!
How about that? And yeah, very nice paper. This could have been TWiV or TWiP, Dickson.
Well, that's why it's a hybrid.
It's a hybrid.
Absolutely.
You got a virus and you got a parasite.
Exactly right. And a bacteria. And you could do the TWiM also.
Boy, this could cover. I could just repost this, right?
Yes, and Thor will be on next week as part of our crossover.
That's right. Exactly. All right, now it's time for a hero, Dickson.
We have a hero. We do. And Peter, in a prior email, sent us the biography of two people. And the person that I will feature in this hero portion of our show was one of the other person's students. And he went on to become a famous malariologist, and his name is Ogobara Duombo, and he's from Mali. In fact, he's from the Dugan region of Mali, which is the upcountry region north of Bamako. And the houses, if you want to see some spectacular examples of native houses that have beautiful designs, and it almost looks like it's a modern design designed by some abstract artist, and he's from that region. Born January 1st, 1956, and unfortunately he died at a young age. According to me, at least, he died at the age of 62 in Marseille, France. He was a medical student in Bamako, and then later on went to France, where he would attend the University of Marseille in France to study parasitology, but for as long as he agreed to return to Mali when he had completed his education. That was the caveat that they asked him to agree to before he started his studies. And of course he agreed wholeheartedly, because he was thoroughly committed to improving the life of people that live in his home country. But he knew that he couldn't get the quality of education that he needed in order to up the level, so to speak, of understanding about a lot of things that he was working on. And he became a researcher in malaria, and I'm sure that Bob Gwads knew him too. He's one of our authors on our book, and a medical entomologist of some note as well. And he ended up at the Department of Medicine at the University of Bamako, where he co-founded and directed the Malaria Research and Training Center. A lot of people sent in accolades of his achievements. I can read one here that I'm thoroughly familiar with because I went to school with him, Dr. Lewis Miller. He's down at the NIH, who heads the malaria cell biology research section at the NIH. He quotes, they were able to define, these two gentlemen, and Duombo was one of them, they were able to define the actual gene that was involved in chloroquine resistance, and that was very important. That was a quote from Dr. Miller. His work was so broad, said Pedro Alonso, the director of the WHO Global Malaria Program. He went all the way from understanding molecular mechanisms to resistance, and his group that really became a leader in clinical trials in the development of antimalarials. All of that happened under his umbrella. He then went on to explore the role of chemoprevention, both in pregnancy as well as in children. The Malaria Research Training Center became an important training ground for Malian medical students and researchers. A lot of his accomplishments were by mentoring younger people who he pushed out to take the lead as first author on papers. So he was a very generous person as well. And it ended up, I'll give you the final quote, his loss is tragic on many grounds for research in Africa. Alonso said at this point, he was in that sweet spot where it was not only about him doing his own research, but about taking global leadership. In recognition of his service, Duombo was given a state funeral by the Malian government. He is survived by his 2 wives and 10 children. Ogobaro Duombo gave a lot to science, to his continent, and to his people. So that's my hero. And he said in the final sentence, his vision and example must continue to each and every one of us to inspire us in our research and in our daily work. So we need more heroes like that, that's for sure.
Very good.
And thank you, Peter, for sending that information.
Yeah, thanks a lot. Thank you, Dickson.
Welcome.
And now...
yes.
Daniel, do you have another case for us?
I do.
No,
I
don't think he should. Don't you think he's out of cases now?
Well, he's got plenty of COVID-19 cases.
Unfortunately. So, no, let's do another. Now, this one I hope is fun and easy, and we get a lot of people writing in, like feeling good about the fact that they know what this is. So let's do the story. As people probably have remembered, our longtime listeners, often when I do cases in a sort of a cluster in different groups, I'll do a couple cases that involve the volunteers, the risks of going to be helpful, and then I move into cases that I see there in the local individual. So this will be the second and last of the risks of being helpful cases. This involves one of those 2 volunteers. So, there's more than one thing can be going on. So now they're better. Now the diarrhea is gone. They're feeling better. And I hate to bother you, but I've got a second issue. Hopefully you can help me. Sure. What is the problem? What could be? Since they came to Uganda to work at this FIMRIC, Foundation International Medical Relief for Children site in eastern Uganda, they have been noticing that when they wake up in the morning, a lot of times they will have a series of not so small, I'm going to say about a centimeter, half an inch or smaller, red raised lesions. And they usually be in a line. There might be 3, there might be 4, and they'll notice them basically on the torso. And they actually get pretty darn swollen, and they're itchy, they're uncomfortable, and basically they've gotten in the habit of they'll inspect themselves before they go to sleep, and when they wake up the next day, there'll be new lesions that have formed. So they start to get pretty annoyed by this. And so finally what they decide to do is they actually talk to the manager who's been there for a while, right? So thinking that he might know. And he says, "Oh, it's such and such. Welcome to Uganda." They are not happy with it. So he recognizes this is a common issue. And he says, he actually, Nick, who I will name, Nick is a Peace Corps volunteer. He's been in Uganda for a number of years, so he had experienced a similar issue. Tells the individual what it is. They're not happy with the answer, so they proceed to move out of that room because they have this idea. This happens when I sleep at night. I'm going to move into a different room. So what they do is they leave all their bedding and they go in and they start sleeping in a different room. They take clothes and other things, they wash them, they go through a whole process, and they stop waking up with those lesions. So a couple questions we have for our listeners. What do we think is going on? And here's the second, which I think is quite interesting. When is it safe for this individual to move back into that room? It seems to somehow be associated with these recurrent skin lesions.
What's the room like? Is it thatched or mud or wood and glass?
So we're living in fancy accommodations. We have concrete floors, concrete walls, a steel roof. Uh-huh. There is about 20, I would say 20, 30 feet from the house to where we have a couple of these drop toilets. There is this was actually a big improvement. So when I was there last, we would fill these buckets, or actually we would hire the local lads to fill these large containers with water, and we would use a bucket and a cup to wash ourselves. But Nick's, I'm beginning to like Nick, because Nick had actually set up some way that we could actually have a real shower where the water, you could turn a knob and water would actually flow down from above. I think called a shower in many parts of the world. But no, the rooms are pretty tight quarters. So it's about 6 foot by about 8 foot bunk beds in most of the rooms, and then an area of wood shelves. You can hang things. All the beds actually have mosquito nets over them.
Mm-hmm. So in these rooms, there's just beds, nothing else. There's no sink.
Nothing. No sink, nothing else. Just a concrete, 4 walls.
And beds.
And beds.
No plants, right?
No plants.
Okay. And this.
No pets.
No pets.
No pets. No animals come in. The windows, do they have screens?
The windows have screens.
Uh-huh. Okay. Are you good, Dickson?
Yep.
All right. Thank you, Daniel.
All right.
Let me read just one email, Daniel, because this is a question for you. It's from Sophia from Greece. Sorry, guys, I have a question from last time that's been itching me.
I like that.
It's a follow-up to this case.
So, if you're infected with worms and get cholera and then typhoid, how does your gut respond to that? And how do you make it through? Isn't it too much? I know I would be dead. Do the parasites make it worse?
So would most other people.
Do the parasites make it worse for someone to get cholera on top, or can cholera affect the worms? Thanks very much. Greetings from Greece.
Yeah, I think this, well, Sophia, I think you're responding to, I had mentioned that a couple months before I got to or returned to the clinic in Uganda, that there had been an outbreak of cholera. We're in an area where there's a large number of people that are infected with intestinal worms. And then we also had some issues with typhoid in this area. It's actually pretty tough. I mean, it's tough to get worms when you're healthy and that's your only problem. It's tough to get cholera when you're already having the impact on your health from worms. Cholera, you're either gonna die or get over, and most people with adequate hydration are gonna get over. Not everyone, unfortunately. Right. So you're probably not going to get cholera and typhoid at the same time, but you might, as we saw in this area, you might have worms. And then you get cholera, and you get through the cholera, and then a period of time goes by, and then boom, then you end up with typhoid. And so you're constantly throwing off your gut microbiome, and the more hits like this, the increased number of people not doing well, and the increased number of people actually dying. I mean, typhoid, interesting enough, is one of those illnesses that I actually was comparing to COVID-19, where people get through it, and the end of that second week, everyone thinks it's all good, and then boom, the ileum perforates, and then the person gets really sick on you. Just comparing that to what we're seeing with COVID-19, people are trucking along, you think it's going to be okay, and then boom, during the second or third week, we see the decompensation. So, yeah, not great to have all these things going on
Nope.
in an individual. You fall down and then you get kicked.
Right.
All right, one last one from Vic. He sends a video for Dickson.
Yes.
Which is Nebraska retiree uses Earth's
heat
to grow oranges in snow.
You can do it.
There you go. Thank you, Vic.
Good stuff.
That's TWiP 181. You can find all the show notes at microbe.tv/TWiP. Of course, you listen on a pod player, which you use on your phone or tablet. And if you do that, please subscribe so you get every episode automatically. And it's free, of course. And we know how many people are listening. We'd love to know that. If you'd like to support us, if you like what we do, consider helping us out financially. Microbe.tv/contribute. You can use Patreon or PayPal to give as little as $1 a month to help spread knowledge for free. Questions and comments, guesses to the cases, TWiP at microbe.tv. Daniel Griffin's at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Pleasure as always.
Dickson Despommier is at Racaniello.org and thelivingriver.org. Thank you, Dickson.
You're quite welcome, Vincent.
See you tomorrow on TWiV.
Yep.
I'm Vincent Racaniello. You can find me at virology.com. I'm going to make it easier for everyone. Because everyone always says, what is that WS?
That's right. It's Western Samoa.
It is, but virology.blog is easier because that's what it is.
That's exactly right.
Could I ask you, Dickson, so people talk about blogging, like video, sort of posting stuff. What is actually the B in blog? Like, where's do you have any idea where this comes from, this term blog?
I do not know.
Anyone?
I was told that once, but I don't remember. We're going to get a lot of letters on this. You know that.
I hope so. I want to know. Like,
it is a truncation of weblog.
Okay, excellent. Thank you.
Yeah. Good deal. The original, we called it weblogs, and then they just said blogs, and that's kind of catchy, right? Blog.
Yeah, I like that.
Right.
All right. Did I say I'm Vincent Racaniello? You can find me at virology.blog.
Yes.
That's right.
Okay. Music on TWiP is by Ronald Jenkins. Thanks to ASM for their support. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.